So everyone in the audience, hello. My name is Jake Montgomery, and you are in the correct room if you're here for this, what's up on the slides right now. And so today we're going to go through some tutorials, and if you haven't already, you could download these GitHub repositories that we have listed here. I think Brent sent out an email yesterday. So you can get the links from there. We anticipated to have a little bit more time at the beginning of our session today to help you walk through that. But I'm just going to get started talking through some background of the first half of this presentation. And as I'm doing that, I'm going to ask you to open up R and to install two packages, just the VCF-R package and QTL2. Brent is responsible for this GAPIT package, and he will walk through that when his time comes. But if you could just open up R, if you're going to follow along today, and run this install.packages command with VCF-R in quotes, and then also QTL2 in quotes. Lucas will be walking around. If you have any issues during this installation step, please flag him down and just talk quietly with him, and he will make sure that you're all set up for whenever we get around to the R portion. So with that, I'm just going to dive in. My name is Jake Montgomery, like I said, and I'm going to be taking the first half of this training. So we're talking about genetic mapping in general, which is basically the process of finding what genes are controlling a trait of interest. And I'm going to be talking about quantitative trait loci mapping, or QTL mapping, which is one of the flavors of genetic mapping. Brent will take over later and talk about genome-wide association studies, or GWAS. And so I want to start by establishing a baseline understanding of what QTL mapping is. Maybe you've read about it or seen it in some publications, but I just want to get everybody on the same page. And so what this is, it is a genome-wide test to identify parts of the genome, physical regions that are controlling your trait of interest. And so if you, for instance, have a herbicide resistance trait that you're studying, and it is because of enhanced metabolism through a P450 enzyme or cytochrome P450, but it's upregulated by a transcription factor, you're going to map to the transcription factor that is transregulating your CYP. You're going to map to the actual thing that is causing your trait of interest. And then you can further study that pathway once you identify it. But this is getting to the actual nucleotides that are affecting your trait. And so this test functions on some assumptions, much like it's basically a big statistical test. And with like a t-test or whatever, you have some assumptions. For that, it's like normality and stuff like that. For this, you need your trait to be heritable. So it needs to be able to be passed on through generations, through genetic material, and it has to be nuclear. So it cannot be like inherited mitochondrially or chloroplastically. You would notice that in your inheritance pattern, like some maternal inheritance, but this is to study nuclearly inherited traits. And so in the last section, I talked about environmental variability. And you want to, the ideal trait for QTL mapping is something that is not very environmentally regulated. So if I come by and I hit your hand with a hammer, you don't have a gene for an inflamed hand. It just happened to you compared to the next person at your table. Similarly, we want to study traits that are strictly genetic and not confounded with environmental situations. And so what are the inputs, and then what do we get out from a QTL study? We put in genotypic and phenotypic information from a segregating population for our trait, and we'll talk more about what a segregating population is in a moment. And then we get out this plot off here to the right. And so this is like the light at the end of the tunnel. If you're doing a QTL analysis, this is what you want in the end. This is a plot of a genome scan. And so I want to explain the axes here for a moment, because this is like the main takeaway of what we're going to finish with, is that you have the whole chromosome listed out on the x-axis, stretch across all of the chromosomes lined up end to end. And then the y-axis is this LOD, this LOD score. It stands for likelihood of odds. And you can think of it as an inverse p-value. So the higher the LOD score, the more significant that point is. And then we have some threshold lines, which we'll talk about how we calculate these, but these are basically 95 and 99% confidence thresholds, to where if you go above that, then we consider it statistically significant that this region is associated with your trait of interest. So you can see here across the whole genome, they only found one QTL at the very beginning of chromosome 9. So now, instead of having to look across the entire genome for what genes might be affecting your trait of interest, you know that whatever it is that's causing it is right there at the beginning of chromosome 9. So now you've narrowed your search from tens of thousands of genes to hopefully 100 or so, maybe less than 100. Okay, so that's why we would conduct that. Now I kind of want to talk through the basic steps of how this actually happens. And we're at a weed genomics conference where one of the most pressing issues is herbicide resistance. Arguably, that's why this consortium was created, was to study non-target site herbicide resistance, along with some other things. So today we're going to walk through a case study that is trying to understand what genes are causing non-target site herbicide resistance. And so the first step, regardless of what trait you're mapping, is to find plants that have variation for your trait of interest. For us, we're talking about herbicide resistance. So this is often done through herbicide resistance screens, where we go out and collect seed from the field or from farmers that are complaining of lack of control in the fields. Then we take it back to the greenhouse or a field experiment where we confirm resistance. And that's a way that we can identify variation. Additionally, if nature isn't doing it quick enough, if we aren't satisfied with natural evolution, we can induce it ourselves through mutagenesis. So we can treat seeds with a mutagen like EMS and then screen the resulting populations to look for phenotypic diversity that we have induced. And then we don't really do this last one, but some breeders might go back and screen diversity panels or land races to identify variation that we've bred out of some crop. Like I said, weeds don't usually need diversity panels to show us some variation. Okay, so once we have identified the variation for our trait, we need to come up with some test to separate out our phenotypes. So for herbicides, we often use a delimiting rate and just spray the plant. This is calculated by a dose response experiment where we take these plants, the sensitive and resistant, then we spray them with multiple rates in the greenhouse, and then we see where the plants, the sensitive plants die and where the resistant plants continue to grow. And that can be a delimiting rate to tell us what is sensitive and what is resistant. Alternatively, Xinji Wu of Bayer recently came out with a really good paper describing leaf disc assays for herbicide diagnostics or herbicide resistance diagnostics. So that's a great way to non-destructively test for resistance in your plants. All you need is a hole punched out of a leaf, not the whole plant itself. And then for the physiologists in the room, conceivably you could also come up with hopefully a high throughput method of identifying variation for metabolism or translocation, how much of the herbicide is getting out of the treated leaf up to the meristem, or how much of it is being metabolized to a non-toxic compound. In theory, that could also be a phenotyping method, although probably a bit more painful and less high throughput than some of the other methods that we've talked about. And so as we talk about population structure here, I want to talk about the figure on the right that I haven't mentioned at all so far. And that is you have a resistant and sensitive parent. And so the genomes here have been reduced to just one chromosome. They have a diploid plant there. it's got 2 copies of just 1 chromosome. In reality, plants have more chromosomes than this, but for the illustration here, we're just going to simplify it down to 1. And I have colored the sensitive to be blue, and that's just to help you track the DNA through these generations. And then the resistant plant will have these red chromosomes. And so when you cross these plants together, which is the main delimiter between QTL mapping and GWAS, in GWAS, you're going to use just natural populations. You're going to go out and sample and conduct your experiment right away. This is a little bit different in that the QTL process involves synthesizing like a synthetic population that you control. So you know the structure of this population. So in the progeny of this cross, the F1 generation, you're going to get 1 copy of DNA from mom and 1 copy from dad. So the sensitive and the resistant parent. And so you can see the phased genomes there. You got everything blue on one strand and then everything red on the other strand. And then, so this F2, which is the resultant of selfing that F1, once you go to the next generation, that's where the magic happens. That's what we're focusing on today. And this is the population that we're going to use for our investigation. So this F2 is important because it is segregating in that some plants will have our trait of interest and some will not. And so the next step in our process here is to collect DNA from all of these F2 plants, usually about 200 or more, and then apply our delimiting assay that we talked about in the second point. So spray them with some rate that you've developed previously that you know will kill your sensitive plants, but not your resistant plants. And that should give you your phenotype data. So I have listed here 3 plants, 3 example F2s. The first one alive, second one dead, third one alive. This is pretty simplistic. In reality, you might take like a measure of biomass or some other metric of survivorship, but this is just to illustrate the point. And so once we have collected the DNA and the phenotype information, then we can go and sequence that DNA to paint these chromosomes and understand which regions were donated by each parent. In the process of gamete formation, you get crossing over. And so the F1, it's completely phased blue and red, but in the F2, you get a mixture of blue and red on each chromosome that is random. And so that is the workhorse of QTL mapping, is that for every other locus in the genome, it's going to recombine and it's going to be totally random there. But for your locus, the resistant sequence will always show up in resistant plants. So that is the smoking gun that resistance leaves that lets us know that part of the genome is responsible. And so once you have the phenotype data and the genotype data, which can be collected through a couple of different sequencing techniques that I won't talk too much about, then you can use the bioinformatic pipeline that we're about to talk about to identify this weirdly thin box. I don't know why I didn't make that thicker, but the box at the bottom of the screen is what we're talking about right here. I guess I can use my mouse a bit more, but so you can see that this region is a candidate region for resistance, where every plant that has red sequence, at least 1 copy, is surviving. But then down here at the very bottom, this plant has blue sequence on both strands, so it's homozygous sensitive, and it is dying. So this is just 3 plants. In reality, you would use maybe 200 or more to get the statistical power, but this is the region that we're looking for. And so anything in this region is a candidate gene for causing your trait. So once you have this region identified, you might feel great satisfaction. I'm sure you will, but you're not quite done there. You need to keep going and at least validate this QTL. So the way that you do that is you make a new F2 population, either with just like the same parent population, just redo it. And then if the QTL that you identified is real, you should be able to discover it again in this newly created F2 generation. So you don't have to do the whole genome or anything, just create one marker for that region and see if it, again, is segregating with your trait of interest. Alternatively, you could jump straight to functional validation. And so this is where we take the gene that we think is causing our trait and we put it in a Arabidopsis, or we knock it out of a Arabidopsis, or grass model like rice or something. But that's kind of getting ahead of ourselves, especially if you have 100 candidates. You're not going to transform 100 or clone 100 genes into a model organism. You'll want to do something to reduce down your QTL size and only move forward with your strongest candidates. Okay, these are the basic steps of mapping. But today we are really just going to focus on the bioinformatic pipeline and how we go from genotype and phenotype to some identified QTL, like some regions of interest. Okay, so I have a plot explaining basically what's going to happen in our statistical analysis in our bioinformatic pipeline here. So we have 8 example individuals here with their genomes stretched across the x-axis. The genomes are painted or colored according to which parent donated the sequence for that locus. So you have a blue parent and an orange parent, and you have recombination events across the genome. So what we're going to do is scan along, and for every marker that we find, we're going to make basically a t-test, or in our case, it's a bit different, but we'll talk about that later. And we're going to compare the mean phenotype of the blue group, so if you have blue sequence, to the mean phenotype of the orange group. And for every non-causative locus, it's going to be flat. There should be no difference in average phenotype value. But as we scan along, you'll see that this line stays pretty much flat. And that corresponds with a low negative log p-value. So this will be equivalent to our LOD score in the end. So that will stay low and this flat until you reach a point here where the mean phenotype value for these two groups is obviously different. And you can calculate an R-squared if you want, but the point is that there's a difference between your phenotype, a difference in your phenotype if you have the blue sequence or the orange sequence. And that is what results in this peak in the LOD score. So here we just found one peak. So we just have to look in this region for genes of interest. Okay, so now we're going to talk about the bioinformatic pipeline a little bit. And this is mostly to lead us up to where we're going to start today. So I'm going to start with the sequence. We're not going to cover the phenotyping method so much. This is more of a genomics conference, so we're going to talk about that a little bit more. But we're going to talk about how you go from raw DNA sequence that you get back from the sequencing core to variants that you're going to send into your QTL pipeline. So we're going to start. We just got our FASTQ file back from the sequencer. We're excited. It's data day, and we're going to get started on the computer. So the first thing you're going to do is go through your FASTQ file and trim your reads. And you're going to do that with this tool, Trim-O-Matic. It just goes through and find errors that we predict in the reads from the sequencer, and it gets rid of them, trims them off. The next thing you're going to do is this step, demultiplexing. So you get back one file from the sequencer, and it has reads from all of the samples that you sent in. But a clever trick that we do is we attach a name tag to the beginning of each piece of DNA that goes to get sequenced. And it says, hello, my name is sample 2, or my name is sample 30. And I'm representing that here with different colors. And so this demultiplexing step, it just separates the reads out. So all of the reads from sample 10 are in their own file. All the reads from sample 30 in their own file. And so that's what this is representing here. The next step is to align these reads to a reference genome. This is the reference genome you can get from the consortium, or there's a few out now. But this is one step that has largely kept us from doing this before, is we didn't have a reference genome to align to. And so this is done with a package called BWA MEM. And so aligning just finds where in the genome our reads came from. It matches the sequence up, and then you can see like this stack of reads. So you have sequence supporting what the sequence is for each sample right here. Once you have these all stacked up, we can call this script GATK to come along and call variants. And so that is to capture, sorry, getting a bunch of stuff popped up. You might have to come and log into this Wi-Fi. That's very sad. This computer just disconnected from Wi-Fi. So did you type in an access code to get on the Wi-Fi? Is it IWGC 2021? Good. That's right there. Or are we gonna have to reconnect to the Zoom meeting? It's working. It just, we need to reconnect to the Zoom meeting. So go to the Zoom. Maybe just let you join. It's fine. What is the passcode?
What is the meeting passcode?
Put us back up on the screen. There we are.
There we go.
Perfect. Okay. So, all right. Just to catch us back up, you get your reads and you trim them, you demultiplex them, you separate them into files, you align them to the genome, and then you find these variants. So these arrows are pointing at sequence variants that exist between samples. The blue sample has a yellow sequence here, the orange does not. So that is what we're going to pass into our QTL package today in R to do our genome scan. And then we're going to finish up by using some command line tools to extract genes within the QTL that we find. Okay, I'm not going to spend too much time on this slide because we already talked about what a VCF file looks like, but this is the file that stores the variants from GATK. I am going to talk very briefly about the input files specific for QTL, the package in R. And so the first one that's going to have all of your summary information is this control file. It gives the names of all of the data files that you'll pass in, as well as some other information. After this training, if you want to look into these files and how to replicate them in your experiments, please do that. But I'm not going to talk too long about this. So the next file is a genotype file. It's derived from our VCF, where we take our variants. But this is for every marker, the individual has the allele status at that locus. So it's going to be SRRS, basically saying whether that sequence was inherited from the sensitive or the resistant parent. And so this is the color that we will use to paint the chromosomes, either orange or blue. Next up is the phenotype file. This is direct data from your phenotyping methods. For us, I'm going to, the simulated data set is supposed to be like dry or fresh biomass 14 days following a herbicide treatment. And so this is in grams here. You could use change in height, plant height, if you're looking at like change in growth or some visual rating or whatever phenotype you want. You can list multiple, each in their own column, and QTL, whenever it goes through, will do a genome scan for each phenotype independently. And then the last one is this genetic map file. The name of each marker seems pretty informative. Chromosome 1, base pair 0. The problem is that QTL doesn't parse this name out because different people have different naming conventions. So you have to tell it explicitly where in the genome this marker exists. So on chromosome 1 at base pair 0 or at mega base pair 1 or so on. Okay, so those are the files that we're going to put in. I'm going to wake everybody up. We're going to get on our studio and actually run some code. So if you have your laptop here or at home and you are ready to follow along, I'm going to ask you to switch over to our studio. Now, okay. Yeah. All right. Thank you. All right. So if you downloaded this repository before last night, you're going to have this top code chunk here. Don't worry about it so much. It's not important. But we're going to start down here. So if you haven't already run this install.packages command, you're going to need to do that. If you have already run it, you can just call this library, this VCFR library that you've already installed. And then we're going to start by importing our VCF file, this Kocha F2 GBS VCF. And we're going to import it into this object called VCF. Next, we can look at what that object contains now. Basically, it's just all of the information from our VCF file. Next, we can extract only the genotype information and then make that into a data frame. And we look at this now, we've just extract the ones and zeros that tell us whether it was the reference allele or the alternate allele at this site. So we're going to write that out to a CSV file. And then we're going to go over to a Python script that I've written. And the importance of this script is that it's going to translate zeros and ones from our VCF file and give us back either R or S. It's going to tell us whether it was inherited from the resistant or sensitive parent. And so the way it does this, I'll run it, but it looks at what the sequence is for the R parent, and then it looks for what the sequence is from the S parent, and then it will go back to each of the samples and use that information to convert to R or S. Okay, so now our genotype file is ready, this Kocha alleles.csv. So we're going to load our QTL2 library and read in the control file. So like I said, this has all of the information for our experiment, all of the input file names, and some other information. So we're going to read that into this object cross, and we're just going to plot out a histogram of the biomass. So you can see this is a pretty normally distributed trait. So assumingly quantitatively inherited, so probably multiple genes affecting this. It's not like a nice 3-to-1 distribution. We have a bell curve shaped distribution. Okay, so The next thing we're going to do is calculate genetic probabilities. This is the only prep work that we have to do before we run our scan. And the reason why we do this is mainly to make up missing data. So it's to impute missing data. If you have one marker that's missing, but you have markers flanking it, you can use those markers to infer what the sequence is in the middle with pretty good certainty. And so we ran this calc genoprob command and saved it in this PR object. And if we look at the names of this object, it's just 2 chromosomes. So I've reduced the genome down for portability's sake and to make it run quickly. This genome only has chromosome 6 and 1. In reality, you would have a name for every chromosome in your species. The dimension names are the names of the markers. So I have one marker at every mega base pair. Or sorry, this is the names of all of the samples. And then this is the names of the markers. So on each chromosome at every mega base pair. Okay. And then if we plot the genome probabilities, we see that the individual number 1 is homozygous for the R alleles up until this point here. And then it has some crossing over event, and then it's heterozygous. So if we change this to 2, you can see it has a couple other crossing over events. But this is what we use to paint those chromosomes like we saw in the GIF earlier. Okay, so now it's time you've all been waiting for. We actually run the genome scan, which is incredibly fast because of how small the data set is. And we can plot this scan and see the output here. So this is the coup de grace of our analysis. This is what we've all been waiting for. We see this one big peak on chromosome 6, but then we see this smaller peak over here on chromosome 1. So how do we know what is truly significant? Like what is statistically significant from this analysis? And so to do that, we're going to use a permutation test. There's no, this is like a parametric way of identifying statistical significance, non-parametric way. And so what a permutation does is it's going to go in and take the phenotype data and randomize it. So it's going to assign a new phenotype to every individual. And so any connection that there was between genotype and phenotype should be completely erased. It should be totally random. And if you do this, and then do a genome scan, the highest LOD score that you find, you know that is due to random chance. And so if you do this 1,000 times and you write down the highest LOD score that you achieve with every permutation, which we will do right now, and then you make a histogram out of that, this is the result here. So this is the LOD score and then the frequency on the y-axis. So 95% of our permutation runs resulted in a peak LOD score of less than this value, about 3 point something. And then 5% exceeded this value. So this is a way of saying we are 95% confident that anything above this line is not due to random chance. So if we look at the summary of this, it gives us thresholds for 95% and 98% confidence. Remember, your permutations are random, just like mine. So you might be getting slightly different threshold scores when you run this code, but they should be pretty similar. So if we save those thresholds and then run this find peaks function, this will show us any identified significant peaks. So on chromosome 1, if we look back up at our scan, from about here to here, from megabase pair 81 to 93, that whole thing is a peak that it identified. And then on chromosome 6, it's centered right around megabase pair 3, which makes sense. It's very peaked right here. And it's very defined. So the resolution is very good for this other QTL. Okay, so now we have these windows, these peaks, And we know that our genetic cause for our trait is in this window. How do we see what genes are in there? So for that, we can turn to the command line, which I just have in R here, and we can run these commands. I have them named something a little different in my computer, but if you are working out in the crowd, this kochia reduced annotations file should be good for you. And hopefully this will, it did not. We had this issue last session as well. It's not recognizing this file in my directory. If you're running it at home, it should work though. So if you run these scripts here, you should get a list that looks something like this of gene names. So for example, this is a predicted gene in that region. It does not look like something that is notably involved in herbicide resistance. So it's something that you might be able to ignore and move on. I simulated this data set to be centered around this chromosome 1 peak has a glutathione S transferase in it. And then this bottom one here has a cytochrome P450. This is meant to show the importance of using other data in your analysis. So if you get a huge list of 200 genes, but you have no idea like anything about the mechanism of resistance, it's going to be hard to reason through those 200 genes. But if you know that your population metabolizes the herbicide, then maybe now you're just looking for only metabolism associated genes or only transcription factors that might be affecting other metabolism genes. And so that's really all I wanted to show here in this R script is you should end up with a list of genes to move on from there. And so I'm going to finish up by moving back to PowerPoint quickly for some closing points. Okay, and so here are some key takeaways that I want you to consider if you're ever designing a QTL experiment of your own, and that is that phenotyping accuracy is essential. Essentially, what you're doing is running a t-test and comparing means. And if you have a bunch of error associated with your phenotyping method, you're not going to get that clear difference in allele groups that you need to identify QTL. Another thing is to use diverse parents. So whenever I showed the GIF or the GIF, every point had its own plot. You can only make a plot if you have a polymorphism between your parents, something that's different, something that's segregating. And so by using diverse parents, you can have more markers and therefore more resolution. In this data set, I only put one marker every megabase pair. In reality, you should have marker density much higher than that. Next up is this point that I've already touched on, is that QTL mapping should always be done with other strategies to understand your resistance or trait of interest so that you know what genes to look for in your candidate gene list. You need something to help you sift through the candidate genes in the end. Otherwise, you're going to be drowning in these gene names that you have no idea why they're there. On that same note, reliable annotations are absolutely essential. So if you get to this region and then you look at the annotation, but the annotation pipeline didn't find the gene that's causing your trait of interest, you're not going to find it. It's just not feasible to scan through with your eyes and try to find like open reading frames or something like that, or try to find genes that were missed. So usually we just look at the annotation file, and if it's not there, then that is very bad for your analysis. Please validate your QTL with an independent F2, or you can jump to functional validation. It's up to you if you think you have a strong enough candidate. And then a last point is that fine mapping is a technique that often follows a lot of QTL experiments to try to reduce down that window size. So you screen a lot more individuals and hope for more recombination events, and that will help you walk in and reduce the size of that window and narrow in on your true genetic element of interest. Okay, so I just want to conclude by saying that all of the data and our markdown file that we walked through today are on GitHub along with these slides. So if you missed anything or had some errors in your code, you can re-download it later or get a hold of me to try to work out why that happened. But with that, I'm going to turn it over to Brent, and we'll take questions collectively at the end of this session. Thank you, guys. Okay, take it away, Brent.
All right, let me get my beautiful face up for everyone to see. Hello, I'm Brent. I will be running the GWAS section of this workshop. Set up. GWAS and QTL mapping sort of go hand in hand. The primary difference between them is essentially, some of the stats, but also the necessity of constructing or developing a population versus simply using the historical recombination events that have occurred within a sample space or a geography. So a quick overview of this presentation. First, I'll do some introductions of the topic and why we should conduct GWAS and when we shouldn't conduct a GWAS, as well as discuss GAPIT, which is the program that I typically use for conducting these statistical approaches. What are the inputs? What are the outputs? And go through a series of 3 tutorial examples describing the effect of various model parameters, characteristics of whatever population you're working with, as well as briefly mention sort of the effects of the phenotyping strategy and sort of the overall experimental design on the end result, and how errors sort of that occur at the beginning of the process, or effectively errors that from the beginning of the process stay with you sort of throughout the whole process. So what is a GWAS? Why conduct a GWAS? And some comments on the design of it. So the goal of a GWAS is to find genetic variation that is associated with phenotypic variation within an unstructured sample set. So here we can see really the 2 requirements. 1, you need genetic variation. If you conduct a GWAS on a set of clones, you're not going to get anything unless you have predominant somoclonal variation, but that's a tangent. If you say develop an experiment screening a new chemistry of herbicide on a population, chemistry is really great, kills everything off. Well, the phenotypes are the same, so there's no phenotypic variation, so you also can't conduct one of these analyses. So you sort of need both of these hand in hand in order to really progress further. How a GWAS works is, instead of developing recombination events through, as one does in the QTL mapping, through taking resistant parent, sensitive parent, crossing them together, generating F2 or subsequent generations, we're relying on historical events where this has occurred. So at the end of the day, you identify specific nucleotides that are statistically associated with your trait of interest. This is not sufficient to indicate causation. Often you're going to identify linkage blocks or a set of nucleotides that have not been broken historically, or at least within your data set, and that your causal variant is probably somewhere in there, or you, of course, found a false positive. Why conduct a GWAS? If you're setting out to identify common genomic regions which control a trait of interest within a large sample space, a GWAS is an appropriate set or statistical approach. For a sample set which approaches the population set, these can turn into reliable markers. So for instance, within plant breeding, for instance, where you know that your breeding pool is of a defined size with known genetics present within it, you're not planning on adding anything more, yeah, you can probably stop, and most people typically do, stop at the GWAS find that linkage block that you want to add to your next variety, make sure it's in your next variety. One of the thing that really draws people towards GWAS as opposed to QTL mapping is that a GWAS only requires a population collection and phenotyping. You don't need to develop a population. Developing a population takes time. Even for some of our weeds, there is a generation time that needs to be completed and progressed. In order to develop the advanced generations, you need to have the structure and the recombination events required for the approach. GWAS, run out to the field, grab a bunch of plants, hopefully with phenotypic information, and you're good to go. When to not conduct a GWAS? Well, one of them I've already mentioned, when you don't really have phenotypic variation for the trait of interest. If everything provides you the same phenotype, there's no variation, there's nothing to correlate with genotype. If you're investigating a rare allele throughout your sample set or throughout your geography, it will get lost depending on how rare the variant is. That variant can get lost through the statistical analysis. GWAS works best when you have the more plants you have, or the more samples that you have that share a causal variant, the better off you're going to be. If you have a low sample size, which is sort of an arbitrary statement, but the more samples you have, normally the better off you are. 200 to 300 is sort of a good sample size to shoot for plants. I've run GWASs with a little over 100 and they've worked. People have ran the GWAS with over 1,000 and it doesn't work, or at least they don't get anything statistically significant out of it. Marker coverage is an experimental design consideration. Essentially, you want sufficient marker density for the linkage disequilibrium that's present within your sample set. If you remember, we're dealing with historical recombination events. So what you're left with is each genome is effectively a series of linkage blocks that have, and each of the blocks have sort of recombined multiple times throughout the ancestry of the individual plants you've collected. If you don't have a marker within each of these blocks, you're going to miss some of the blocks, which means you're missing some of the genome. And if your luck is as good as mine, your causative marker or your causative variant is probably in one of the lost linkage blocks. So you typically need to use a higher density sequencing approach compared to something like a linkage map, where you have your recombination events, where you have larger linkage blocks. Gaffit is a program written in R that is nice, short, friendly, easy to use, makes a lot of pretty graphs. They routinely update it when a new GWAS model comes online. They typically add it in short order. What it does is it implements multiple different statistical models for genome-wide association study, as well as genomic prediction. I won't talk about genomic prediction in this section, but it may be relevant at some point to some people. One of the nice things, aside from making very pretty graphs, about Gappit is it can produce most of the required model inputs internally. So if you think of your traditional mixed linear model, it will typically have some measure of population structure and some measure of kinship incorporated within the model, and those can be produced by Gappit from the genotype input file. The only two inputs that are required for Gappit to actually run is phenotypic data. So typically in rows, you will have, or in your rows, you will have the samples present within your data set, the sample ID corresponding to the field in your genotype file, as well as distinct columns for phenotypes. Missing data is allowed, so say APAR9, you undergrad didn't water the plants and it died from dehydration stress or something like that. You can just put NA in the field. and GAPIT will just pluck out that phenotype when there's a missing data present within there. Genotypic data is typically stored as a HapMap or a numeric file. That's the file formats that GAPIT takes as an input. It's essentially you need to convert a VCF file, which has been discussed previously, to this format. Tassel, for instance, will have a quick script that'll just switch the formats. They contain the same information, they just have a different representation, and the people who made GAPIT are also a big fan of Tassel, and they made it much more compatible with Tassel than with anything else. There you can impute missing genotypes. Genotyping technology isn't perfect. You're going to have regions of a specific sample at a specific marker information that just didn't get sequenced for whatever reason. You can use an imputation strategy, which essentially, well, the better ones will take highly related individuals to the missing information, and based off that, make its best guess for what genotype should be put in. And so going to our briefly, where our studio, share script. This is the R script that I shared with everyone. We have loading GAPIT. They never put it on the CRAN depository. They keep updating it, and so it's never really in a stable format to push it there. So this is a website download. If you Google GAPIT, you can find their user manual, and there's also how to get it off of GitHub. That requires dev tools. Dev tools can take a while to install as a package. This here is a quick check. So, oh, that's Control+. Okay, you don't want to do that. There we go. Hopefully you can all see now. Install your package, set your working directory to wherever you have the data downloaded to, read in your phenotype file, read in your genotype file, then we can do some making of directories, move to a new directory, because GAPIT is going to produce a lot of output. And we can press the, what I like to call the run GWAS button, which is essentially calling GAPIT with all of the different parameters that you want to call. This line, I can go parameters. Y is essentially the phenotypic variables that you care about. In this case, a sample name, which is important and tells you which sample or which phenotypes map to which genotypes. The data set for tutorial 1, genotypic information, which I had previously converted to a different format, so it runs a bit faster. Principal components for the models that have a Q term and the different models that we are going to run. So we click that, we press run, and it starts running, which means I get to stop sharing that and I get to move back to my PowerPoint to talk about what I just read. Genotypic data stored in a HapMap or a numeric format, simple conversion, not too hard to do. Impute missing data if present makes it run a lot happier. Right through that. So as tutorial 1 is sort of running, I'll talk a little bit about this. In this tutorial, we are looking at the effects of different statistical models. So GLM, MLM, from CPU and link, as well as the effect of minor allele frequency. GAPIT will, as part of its output, produce a number of pretty looking graphs with various amounts of information. I've listed them here in sort of brief descriptions, but I can go over them with actual examples. Manhattan plots are the ones everyone likes to look at because that shows you how many variable or how many nucleotides are statistically significant in a nice graphical format. Association table is essentially a table format of the Manhattan plot, and it also can estimate heritability. First thing you should probably look at is the phenotypic view. Make sure you don't have any major anomalies within your data set. You want a distribution of phenotypic values to be observed, though you can do sort of case control studies where you have essentially a qualitative measure. But what you want is to have some samples on one end of the distribution, some samples on the other end of the distribution, and some samples in the middle. It's that variation in phenotypic values measured, which is going to give you your power and your ability to detect significant associations. So we look, we have, if you sort of squint your eyes, tilt your head, we can see it's sort of, potentially normal distribution. At a certain sample size, it doesn't matter so much. Marker distribution versus linkage disequilibrium. Essentially, this is a check to see if we have a sufficient marker coverage or if we're potentially missing areas of the genome. For the marker density plot, we have distance between marker A and the subsequent marker, as well as the frequency of things in that bin. It's a histogram. What we want to see is that most of the markers are fairly close to the other markers, and that the percentage of the markers, percent accumulation of all of the markers is largely close to 0 or within a small number. And that shows that you have a high marker coverage. The second plot, which is looking at LD decay, sort of describes linkage blocks effectively present within your data set. If we see a high correlation at a higher distance of marker separation, that means those two markers are associated over a longer distance, which means you probably have a linkage block. Markers within a linkage block will tend to provide the same answer, so you can do some data filtration based off of linkage disequilibrium to reduce your sample size, and that effectively reduces the multiple testing correction that needs to be done, since we're normally doing tens of thousands to hundreds of thousands of simultaneous statistical tests, which you have to then correct through a false discovery rate or some sort of other statistical correction. Measures of heterozygosity. So a bit about the data set. This is a publicly available Palmer Amaranth data set taken from one of Todd Gaines's papers of wild collections of Palmer Amaranth dioecious species. We know we have high heterozygosity, so we expect, individuals have some level of heterozygosity, and markers tend to be heterozygous. If you're dealing with an inbred population, you would expect low levels of heterozygosity in individuals, low levels of heterozygosity in markers. And essentially, if you have an inbred population and you see high heterozygosity, you probably have a problem. Population structure is Q term within mixed linear models as well as general linear models. This is a simple PCA that Gappit runs internally. You can see each additional principal component included accounts for reduced amount of the total variation present within the data set. This is showing a bit of the separation of PC1 and PC2, and we can see that, yes, there is some level of population structure present within population or the sample set, and the principal components are separating them out to some degree. Kinship instead is a pairwise comparison between each sample and every other sample. In this graph, red is essentially highly related. Yellow to white is essentially not very related, not related at all. And what we can see is that subsets of the samples have a higher relation to each other than to other samples. And this shows that there's this graph show that there is some level of relatedness between the samples, as well as some level of population structure present within the data set. And therefore, we should probably account for those in the models that we tend to run. Sort of the manifestation of this can be seen sort of in this QQ plot. GLM model only uses your Q term, so your PCA output. Mixed linear model is a KQ model, which includes the kinship as well as the population structure. If we look at the GLM, for the QQ plot, we can see it deviates quite quickly from our expected line, which is probably not good, and we expect to see an inflation of the p-values present when we run the test using this model. Therefore, we should probably select a different model. Mixed linear model closely follows with the expected line until we start to look at sort of very extreme p-values, which start to deviate, which is much more of what we expect. Moving to our two other models, we see a similar thing to the mixed linear model. Another thing to consider when you're selecting your model is sort of the ratio of true positives and false positives and false negatives as well. This can really be measured through some sort of a simulation. The test data set that I provided is one instance of one simulation, essentially. And when we look at this one instance, this one simulation, and compare our different models, we can see, if you look at the output, we can see that only two of our models identified all three of our true positives, in the case of mixed linear model, also several linked alleles, but no, they didn't provide any false positives. And that's sort of what you want. You don't want to choose a model which within the structure of your data set will produce a lot of false positives. And I will stop sharing that screen and I check to see how my RStudio ran. All right. Let me share my RStudio. So we can see in the console that RStudio ran a whole bunch of stuff, gave us a whole bunch of updates and a whole bunch of warnings, but those aren't important, so ignore them. If we look at our file directory, we go to the folder that we created, we can see a number of different file outputs. Some of these are the files that I've sort of discussed before. We have our Manhattan plots, we have our phenotypic view, we have our QQ plots done for each of our different models. So farm CPU, GLM, and it just goes through all of the different models. What I want to find, I think, is why is my brain slipping here? I called it the association table. Results, there we go. View file. See if that loads. That does not load very well. Okay. Stop sharing that. Open it up in a good old program like Excel. Mixed linear model, GWAS output. Zoom that in so we can actually see, and I will share this screen. This is the association table, sort of as the output. We have the SNP name, position, chromosome number, unadjusted p-value, minor allele frequency, important statistical stuff, corrected p-value, and approximate effect size. So if we just take a look at our top 3 statistical hits, it's sorted based off the FDR adjusted p-value. We can sort of look at, so to describe the simulation briefly, I put 3 QTN present within the data set. Each of the QTN had the same effect size as well as the same error that was associated with it. The only difference between them is the minor allele frequency. So this is to illustrate the effect of minor allele frequency. What we can see here is the scaffold 5 QTN had a minor allele frequency of 0.46. So almost half of the plants within the data set had this allele that could be identified. And as a result, we had a fairly high corrected p-value, but as we reduce minor allele frequency, even though it has the same biological effect size, we see a reduction in effectively the p-value. And if you reduce the number of samples down to a certain level, a certain rarity within your data set, it will eventually disappear because you simply don't have enough cases for your statistical test. That goes into when you're starting to look for rare alleles. GWAS may not be the approach you want to use unless you have some way of enriching your samples for that rare allele. Enriching your sample set and skewing your sample set such that you have a high number of the alleles you want to further investigate versus not. But that requires information most people don't bother to generate. Second tutorial, looking at trait heritability. One, you don't really have too much of a ability to fix. Heritability of a trait is dependent on the population and sort of the phenotype that you are measuring. Sometimes we're interested in low heritability traits. Sometimes we're interested in higher heritability traits. Higher the heritability, in general, the fewer samples and the easier it is to detect associations with. And that's sort of demonstrated here in tutorial 2. We make ourselves a new directory, set it to our output directory, and we once again press the GWAS button using the mixed linear model because it runs relatively fast, and from our previous simulation, it seemed to work well within the data set. I won't go too much and look at the individual files, but this is a brief summary. Once again, it has a fairly similar setup to before. Here I have 3 scenarios. One scenario where we have a medium heritable trait, a low heritable trait, and a very low heritability trait. One of the things that this on the pie chart on the side are showing the proportion of the total variation that could be put into your genetics or to everything that's not genetic. So it's a measure of heritability. As you reduce the heritability of your trait, what you see, first of all, is p-values get lower, and once you get to sufficiently low heritability, you start losing your abilities to detect alleles unless you start increasing your sample size. Third and final tutorial I will talk about is the phenotyping strategy. So this is something that we can actually have some influence on. We're the ones deciding how we're going to phenotype our given plants. For this scenario, all of the phenotypes are derived from the same simulations. All alleles have the same effect size. what I did was I calculated effectively the true phenotypic value for the samples plus a given error term. That's effectively your heritability, same thing as our previous tutorials. Then what I did was I simulated what would happen with a delimiting rate screen, where you're taking a diverse population, you're screening at a single rate, collecting, let's say, biomass after 2 weeks or so. What we would expect to see under this scenario is you would have a range of phenotypic values if you selected a good delimiting rate. But once we start to get to the extreme end, say once we reach a rate that's lethal to the plant, we can't differentiate a sensitive plant from an even more sensitive plant. They would end up with the same phenotypic values. Same thing with a resistant and a very resistant plant. So as a result, I binned values up at the extreme edges of the phenotypic distribution. The final one, which phenotypically is it's the easiest one to phenotype, but may not be the best, but maybe the least informative, is sort of a survivalship. You take a delimiting rate, you spray your population, you say this plant lived, this plant died, and you don't take any other measures beyond that. Simple. If it's sufficiently sensitive, it died. If it's sufficiently resistant, it survived. Once again, code to run the GWAS, press the GWAS button, same thing as before, make a new directory so it puts the output in a different folder, and look at the result. The moral of the story for this scenario is that actually quite interesting. As we reduce the accuracy of our phenotyping, we effectively reduce the heritability of the trait. So when we have the true dose response, we have the highest heritability with about 30% of the variation for the trait being explained by genetics. But when we just do the simple survivorship, we're down to 23. And for this specific simulation, as it happens, we even lost one of our QTN in the analysis. We weren't able to identify this QTN on scaffold 1 as a result of the looser phenotyping. So this just goes to show that take some consideration into your phenotyping. The more precise you can make it and the less error you introduce through the phenotyping and through the other stages of your experiments before the GWAS, the happier you are going to be, at least in terms of getting statistical results to continue to examine and move forward. I think that is my last slide. So you guys can all look at my glorious face again, and we can take any questions if you have any on this section.